Morning students. Today you see the topic Escherichia coli. So Escherichia coli, better known as E. coli, are common and derives group of bacteria found in food and the environments, and they are intestines of the both peoples and certain warm blooded animals. So the name of the genus is given after Escherichia coli, who first isolated the bacillus from feces of infants suffering from diarrhea. So they have name Bacterium coli, common, the 1885 to organism. The unlike other coliforms, E. coli is the parasites inhibiting only in human and animal intestines. So morphology of E. coli. So E. coli is a gram-negative rod-shaped organism. It is 1 to 3 and 0.4 to 0.7 micromillimeter in size and 0.6 to 0.7 micromillimeter in volume. It is arranged in singly or pair. It is motile due to the peritracheal flagella structure formation. Some strains are a non-motile organism. So they are non-sporing, non-capsulated. They are facultative anaerobic organism and growing across over a wide ranges of the temperature of 15 to 45 degrees Celsius. Antigenic structures. The heat-stable lipopolysaccharide formation is the major cell wall antigens of E. coli. The E. coli possesses the 4 antigens: H antigen, O antigen, K antigen, and F antigen. First one, H or a flagella antigen. So the flagella antigen is a heat and alcoholic labeled protein formation. The present on the flagella. So the genus specific and present as a monophoric structure formation, and scientific H antigen have been recognized. Next one, O or a somatic antigen. The heat stable resistant to a boiling up to 2 hours. So 2 hours to 30 minutes. And occurs on the surface of the outer membranes and integral part of the cell wall structure formation, and 173 O antigen has been recognized, the somatic antigen. Next one, K or a capsular antigen. The heat stable and acidic polysaccharide antigen present in the envelope structure formation. The boiling removes the K antigens. So it is a inhibit the phagocytosis. So 103 K antigen have been recognized. Next one, F or a Fimbria antigens. So it is a heat labeled protein formation. The present in the Fimbriae, and K88 to K99 antigens present. Next, cultural characteristics of E. coli. So the Escherichia coli is a facultative anaerobic organism. Its optimum growth of temperature is 37 degrees Celsius, ranges from 10 degrees Celsius to 40 degrees Celsius. So E. coli on a nutrient agar, they are appear large, circular, low convex, grassy, white and moist, smooth and efficacious structure formation. There are two form of colonies: smooth form of and rough formation. Smooth S form and rough is R forms. The smooth forms are emulsified in salines. So easy kadaiyakudiya thanmai udiyathu. So due to the repeated subculture, there is smooth to the rough various S and R variations. So E. coli on a blood agar medium, the colonies are big, circular, grey and moist. It's a non-hemolytic colonies. So gamma hemolysis examples or a beta hemolytic colonies are formed. So next one, E. coli on a mechanic agar medium. So the medium, the colonies are a circular, moist, smooth and end their margin structure formation. So the colonies appear flat and pink color colony formation. They are lactose fermenting colonies. Next one, E. coli on a EMB agar medium. So the green metallic shines colonies are formed EMB medium. So E is in methylene blue agar. So next one, E. coli on a liquid media. There shows homogeneous turbidity growth within 12 to 18 hours. So R form agglutination spontaneously, the forming a sedimentation on the bottom of the test tubes. The after prolonged the incubation less than 72 hours. So the pellicle are formed on the surface of the liquid media. So next one, biochemical characteristics of E. coli. The E. coli difficultly produce the positive test result for indole, lactose and mannitol, and fermented as produces a gas form gas from glucose formation. So virulent factors of E. coli. So the E. coli passes a broad ranges of virulent factors. In addition to the general factors passed by all membranes of the family, this means the Enterobacteriaceae. So the E. coli is a strains passes the specialized virulence factors that can be placed into two general categories. First one, adhesion and exotoxin. So enterotoxin E. coli, the colonies factors antigens, the colonies factors antigen 1, the colonies factors antigen 2 and colonies factors antigen 3. There are 3 types of enterotoxinic E. coli. It's a heat labeled toxin LT1 and heat stable toxin STA. And enteropathogenic E. coli, it's a bundle forming pili, it means. So enteroaggregative E. coli. So it is aggregative adhesions fimbriae, adhesion aggregative fimbriae 1 and 2 and 3. So the enteroaggregative is the heat stable toxin and plasmid encoded toxin, those are produced. The Shiga toxin producing E. coli, STEC, so bundle forming pilis and intimates. The Shiga toxins 1 and 2, those are produced. And next one, invasive E. coli. So invasive plasmid antigen and hemolysis formation. So next, uropathogenesis, the pili, P. pili and DR fimbriae formation. So pathogenicity of E. coli, the majority of E. coli infections are opportunistic infection. The E. coli is the most prevalent cause of nosocomial infection. The diarrheogenic E. coli strains passes the adhesions to produce the toxins that are responsible for the development of diarrhea. And first one, the E. coli causes the different types of E. coli infection. First one, opportunistic infection. So E. coli organisms are found in large numbers in human feces and are spread via fecal contamination. They are frequently of a nosocomial E. coli. The infection increase when invasive producers such as the catheterization are used. So next one, urinary tract infection. So the community acquired urinary tract infections are usually the result of cell infections, occurs most often in worm. The other risk factors for urinary tract infections include the urinary catheterization, sexual intercourses, diabetics and the presence of kidney stone formation. And last one, the diarrheal disease. At least 5 groups of E. coli causes diarrhea. So clinical feature of E. coli. So gastroenteritis, so enterotoxinic E. coli causes a traveler's diarrhea or infant diarrhea in infant. The pathogenesis involved the plasmid mediated, it's a heat stable and heat labeled formation, enterotoxin that stimulate the hyper secretions of fluids and electrolysis reaction. Enteropathogenic E. coli causes infant diarrhea. In developing countries, the pathogens involve the plasmid mediated and histopathology will disturb of a normal microvilli structures resulting in a malabsorption and a diarrhea. So enteroaggregative E. coli causes infant diarrhea in a developing and probably developed countries along with the traveler's diarrhea. The pathogenesis involved the plasmid mediated aggregative adhesions of rods, stacked and bricks with a shorting, a microvilli mononuclear infiltration and a hemorrhagic and decrease the fluid absorption formation. So next, STEC, the causes the hemorrhagic colitis. The STEC, the annual the form EPEC, enteropathogenic E. coli. So the lesions with the destructions of intestinal microvilli resulting in a decreased absorption pathology mediated by the cytotoxic Shigella toxin. So it is a Shigella 1, 2 and 2 which is disrupt the protein synthesis formation. So next one, invasive E. coli, the causes the diarrhea which is rare in developing and developed the countries. The pathogens involve the plasmid mediated invasions and the disruptions of epithelial cells leading to the colon. So next one, urinary tract infection. So the most gram-negative rod shaped that produce the urinary tract infections, the originated the colon, contaminated the urethra, ascended into the bladder, and may migrate to the kidney area prostate formation. So although most strains of E. coli can produce the urinary tract infections, the disease is more common with the certain specific serogroup. Sepsis formation. So when a normal host defense area inadequate E. coli may reach the bloodstream and causes a sepsis formation. Newborns may highly susceptible to E. coli sepsis because they lack IgM antibodies. The sepsis may occur secondary to a urinary tract infection causes. Next one, meningitis. The E. coli and group B streptococci are the leading causes of meningitis in infant. So approximately 75% of the E. coli meningitis cases have the K1 antigen. This antigen cross-react with the group of B capsular polysaccharide of N meningitis. The mechanism of virulence associated with the K1 antigen is not understood. So clinical manifestations of E. coli. It's produce a gastroenteritis formation, watery or a bloody diarrhea, vomiting, cramps, nausea, low-grade fever, dehydration, abdominal cramps formation. So the urinary tract infections, the most common bacteria found to the causes urinary tract infection is Escherichia coli. The other bacteria can cause a UTI, but E. coli is the culprit about the 90% of the time. The major manifestations of the infection include a strong persistence, urinate, and burning sensation with the urinating, and pelvic pressure, low abdominal discomfort, and frequent the painful urination and blood in urine. Lab diagnosis, laboratory diagnosis of E. coli. So the urinary tract infections, most urine specimens are obtained from adult pattern via the clean catch midstream urine techniques. So the bacteria can be detected microscopically using the gram staining of a uncentrifuged urine specimens. The gram stains of centrifuged specimens are a direct observations of urine or bacteria in the urine specimens. On staining, E. coli appear on a non-sporing formation. So the gram-negative rod-shaped bacteria, the routine urine culture should be plated using the calibrate loops for a semi-quantitative methods. Opportunistic infection. The E. coli is a commonest causes of a nosocomial pneumonia in patients who are over the 50 years of age and have a chronic underlying disease formation. So the wound or bowel surgeries may lead to the E. coli wound infection or post-operative infections. Though the infections are diagnosed by a culturing E. coli from blood or samples of the infected tissues. The next samples, urinary tract infection. The samples, the patient may develop the E. coli infection of the urinary bladder, cystitis, or other renal parenchyma and a pelvic. The urinary tract infections are diagnosed by testing and culturing in a midstream urine sample. The urine of patient with cystitis containing erythrocytes, leukocytes, and bacteria. The urine of patients with polynephritis containing erythrocytosis and cast and bacteria that antibody coated and can be detected via fluorescent antibody test. So diarrheal disease. The most patients of E. coli infection causes a watery diarrhea. So diarrhea, nausea, abdominal cramps, and low grade fever extra. And E. coli diarrhea is diagnosed by the isolating the organism in culture and then using specialized test to identify the responsible strain. The exams including the DNA probes and bioassay for enterotoxinic E. coli and tissue cultures test for the invasive E. coli and bioassay for hemorrhagic E. coli and serotypes of enteropathogenic E. coli. So preventions of E. coli infection. Will there isn't vaccines or medication that can keep you from getting sick from the E. coli, there are steps how you can take reduce the risk. First one, wash your hand. So always clean your hands well after getting to a bathroom, changing a disappear and touching animals at home and a place like to zoo. It's also important to wash up before preparing or eating a meal. So next one prevents cross contamination. So always wash hands cutting boards and utensils and kitchens countries with the hot soaps water after they come in contact with the raw meat, and always keep raw meat and fish produce the separate. So the cook meat well. They use the meat thermometers to make the sure meat is cooked at least 160 degrees Celsius Fahrenheit, 70 degrees Celsius. Don't weigh on the color of the meat, as that's not always a reliable indicator of donates. So drinking pasteurized milk. So they avoid any unpasteurized drink, dairy product and unpasteurized juices. Treatments of E. coli infection. The sulfonamide, ampicillin, cephalosporins and fluoroquinolones and amyloglycins have a marked antibacterial effect against the entities, but variations in susceptibility is great, and labor tests for antibiotic susceptibility are essential. So E. coli meningitis record the antibiotics such as third generation cephalosporin, ceftriaxone. E. coli pneumonia record the respiratory support. The adequate oxygenation and antibiotics such as the third generation cephalosporin and fluoroquinolone is the best antibiotic. Thank you.